Today it's extraordinarily rainy, so it's maybe not so nice today. It is my pleasure to talk to you today about multisensory integration and its principles. And we're going to move from sort of a brief review there to talking about multisensory development and the importance of multisensory experience in that development. And from that, we'll segue on to talk about adult plasticity and multisensory integration, just very briefly, because I want to get to this translational application that we've been working on, where we're using multisensory training to rehabilitate a visual disorder called hemianopia. So just to get started, these are iconic representations of the classic 5 senses. So we've got vision, audition, somatosensation, gustation, olfaction. And everybody knows that we have multiple senses. What not everybody has thought about is how interesting it is that each one of these senses transduces a different form of physical energy. So, for example, vision is transducing photons that are emitted by or reflected by objects in the environment, and audition is transducing pressure waves that are traveling through a physical medium, typically air, but we can also hear underwater. And then somatosensation, there's a variety of different types of receptors in the skin that are responsive to different types of touches, etc. And some people might add a few senses to the classic 5, for example, nociception or balance, proprioception, etc. But the key here to recognize is that you're talking about the transduction of different forms of physical energy that have nothing to do with one another. And these different energies transduced into signals that go and are processed to different areas of the brain. So these areas are largely unisensory and generate for us kind of unique qualia associated with each one of these senses. So there are a lot of advantages to having multiple senses because we live in a multisensory world. So one of the advantages of having multiple senses is that they can compensate for one another when one or more is compromised. So we've all had this experience of trying to navigate a dark room. So vision is compromised. What do? You sort of feel your way through it using somatosensation to substitute for your compromised vision. You might have also had the experience of being in a noisy crowd celebrating a win. And in that sort of environment, your audition is compromised. And so if imagine yourself to be in this crowd and you're trying to indicate to your friend that, we should go to that restaurant over there, what do? You start using hand signals. You start using visual cues to substitute for what you would normally do, provide auditory cues to communicate that information. But the benefits of multisensory integration aren't just limited to these kind of extreme example cases of sensory substitution. But because we live in a world, it provides us with a lot of cues to which our individual sensory apparatuses are sensitive, right? We are constantly being bombarded with stimulus information, sensory information, and a lot of that sensory information that's being registered by different senses is derived from common events. So if we imagine ourselves to be in this busy city street, we can see the cars and we can hear the cars. And what our brain does is takes that information about the car that's registered by the different senses and puts it together to provide us with really powerful benefits in perception and ultimately decision making. Okay. So you don't have to take my word for it. This is a clip that I pulled from Sanjay Gupta, who was CNN's medical correspondent, and he was actually up for Surgeon General, I think, back in the Obama years. So this is a clip I pulled from him, and he's talking about,
for example, according to the study.
He's talking about a study that was studying multisensory integration, talking about the benefits of multisensory integration. So here we go.
I can tell.
Let's do this again. Here we go.
For example, according to the study, they had someone talking, just saying these single syllable words in a moderately noisy environment. And they found that the person who was hearing it only got it right 10% of the time. Now, if they heard it and also saw the person in that same environment, they got it right 60% of the time. So it went up 6-fold if you could simply be sitting face to face or across like we are right now.
So he's talking about a study by our friends Lars Ross and John Fox, who are at Rochester, and it's just showing you this massive enhancement in speech perception that you get by watching somebody's mouth as they're talking. Even if you're not a lip reader, right? Just having both sources of sensory information, your brain is integrating that information and producing this massive enhancement. It's a very nice study. So let me show you what he says later on in this clip.
I can tell you as a neurosurgeon, there's only really one area of the brain that processes both seeing and hearing together. Otherwise, when you see something, it's processed in a different area of the brain versus where you hear things. But there's one area called the superior colliculus, which you could use in Scrabble if you'd like, and that's an area where you integrate both the audio and the video together.
So that's not exactly correct. There's lots of different multisensory areas of the brain that integrate visual and auditory cues. And in the speech perception study, it's probably not the superior colliculus. But the reason that that's said is because the superior colliculus has been such a successful model for studying multisensory integration over the years. So let's talk a little bit about the superior colliculus, where it is in the brain, what it does, and how it integrates cues across different sensory modalities. Now, for many of you, this is just going to be a review, but in case there's any students in the audience or something like that, then this might be new information from them, for them. So here is the superior colliculus in a cutaway of a cat brain. Okay, it's bilateral structure. It's got superficial and deep layers, and what I'm going to be talking about today are primarily the intermediate and the deep layers. Okay, so it's located here in the midbrain of the cat. It's called superior colliculus in mammals. It's known as the optic tectum in avians and reptiles and amphibians. And its functional role is to help animals detect and localize and orient to salient events in the environment. So here we have a cat who's detecting a mouse. So the superficial layers of the SC are purely visual, but if we move into the intermediate and deep layers, we can find sensory representations of space for multiple sensory modalities. So there's a topographic map of visual space, but also a topographic map of auditory space, and even a topographic map of somatosensory space, which is kind of funny to think about. But basically, it's like the face is in the center, and then as we move down the flank, we move into the periphery of a somatotopic map. Okay, so if you're doing sensory recordings across the SC, you can find these topographic maps of sensory space, and they're in alignment. So what you're really looking at in the intermediate and deep layers of the SC is a multisensory map. And what that means is that you can identify individual neurons in the deep layers of the SC that are responsive to multiple sensory modalities. So here, just for example, we have a visual auditory neuron. It's responsive to vision and audition. And what that means is that because it's embedded in a map, we can identify regions of space to which the neuron is responsive to visual cues or to auditory cues. And we call these its receptive fields. You can map these out in a variety of ways. And then we can test the neuron with visual stimuli presented alone. And when we present a visual stimulus within its visual receptive field, we'll drive some response from that neuron. And this is an impulse raster. Time is the x-axis. Y-axis is different stimulus presentations, so we call trials. And we can just kind of count up how many impulses the stimulus is producing and call that the visual response magnitude. And that's just plotted here in summary form down in this bar graph. So this neuron produces like 6 impulses in response to this visual cue. So presenting the visual stimulus gives us a visual response. We can present an auditory stimulus within the auditory receptive field and drive an auditory response. So here you see it's a little bit weaker than the visual. And we can even sort of plot a sum line here if we wanted to do that. And then we can present the visual and auditory stimuli together in spatiotemporal congruence and get a multisensory response out of the neuron. And so you see that it's much more robust than either of the 2 unisensory responses. And in fact, in this case, it's actually greater than the sum. We typically quantify that amplification in the multisensory response relative to the unisensory responses with a metric called MSI or ME, Multisensory Integration Index, or multisensory enhancement. It's just kind of a gauge of the benefit of multisensory integration at the physiological level from the perspective of a single neuron. So, like I said, the SC facilitates animals' detection, localization, and orientation to salient events in the environment. And so you might be wondering, well, is there a behavioral assay of this sort of physiological enhancement that you just showed me? And there is. So we've got a nice, robust assay that asks animals to detect, localize, and orient to visual and auditory stimuli that randomly presented from LEDs and speakers that are in a perimetry apparatus that looks just like this. Okay, so we can train animals to localize visual cues or localize auditory cues presented at random locations. So there's a visual cue, so you train the animal to go to that. Here's an auditory cue, you train an animal to go to that, and they're presented at random locations. And then what you do is you reduce the intensities of those cues to make the task hard. Okay, and then you test the animal with the visual and auditory cues presented alone or together to get some measure of any multisensory benefit they might be getting. So here are some data showing you the results of this task, showing you visual and auditory response accuracy at different locations in space. Again, it's lowered because we've lowered the intensity of the cues, right? And then in purple, you see what you get in terms of accuracy when the visual and auditory stimuli are presented together. And you see the same, almost the same magnitude of enhancement that I just showed you in physiology, right? Nice big bump, okay? So that also helps orient you to some of the future slides in which I'll be showing you circumstances where that bump doesn't exist, okay? So now you have seen what multisensory enhancement looks like. We'll talk in a little bit about some cases in which multisensory enhancement is compromised. All right, so it's not the case that just any old visual cue and any old auditory cue gives you these enhanced responses. Rather, computation in the superior colliculus follows, multisensory computation follows a set of principles, and these are well known and sort of well established in the field. Up here in the upper left quadrant, I'm sort of showing you the classic enhancement that you get when you present cues together in space and time. So we call that spatiotemporally congruent or concordant. If you move the cues apart in space, still presenting them simultaneously, and so from a neuron's perspective, moving them apart in space means you move one outside the receptive field. From an animal's perspective, it literally means just separating them in space. If you do that and present them simultaneously, you'll actually get a response depression, as if there's a competition happening between the cues. And maybe that makes sense because it's a structure that is oriented towards, its detection, localization, and orientation. If you have two different cues, it's as if they're coming from different bins that are sort of competing for an orientation response. Okay, so this is called the spatial principle of multisensory integration. Spatially disparate cues typically give you depression. And there's a temporal principle of multisensory integration illustrated in the bottom left. And this is kind of the same thing. If you present the cues at the same spatial location, but separated in time by a lot, then like 300 milliseconds or so, then you don't get significant enhancement. You don't get enhancement above the best unisensory response like you should. And then the final principle here is well established. It's in the bottom right quadrant. This is kind of simple. If you increase the effectiveness of the cues, then the proportionate enhancement you get from their combination is reduced. So that's called inverse effectiveness, but it's just kind of common sense too, because as cues move to very high efficacy levels, or as animals' accuracy goes to like 100% with an individual modality, then obviously the benefit you're going to get by using another modality is going to drop. Okay, so we've done a lot of work over the years identifying these principles by which neurons and animals are integrating cross-modal signals. So the visual auditory signals, et cetera. And we've worked out a lot of the computational principles, and what's illustrated over there on the right are kind of two neural network models that we've developed. One is a single neuron model, the other one is kind of a larger circuit. And you can kind of look those up, and if you're interested in that sort of thing. What we found from a lot of these studies is that the signals that are coming down to the SC that are cross-modal, right? So the SC is a primary side of convergence. The signals that are coming in are unisensory. And those signals are coming in and they're being transformed into a multisensory output in sort of real time. So it's not like the system waits and figures out there's a complicated circuit computation or something like that before you start seeing enhanced output. Rather, you're seeing streams of information coming into the SC, and it's being transformed into a multisensory output sort of in real time. So these streams of information are coming in, and they're converging onto a single neuron and undergoing this sort of churning transformation. So when you tell people that, sometimes they think, well, okay, but am I looking at anything special here? Or is multisensory integration just the result of convergence? Okay, so am I getting amplified responses, multisensory responses out of these neurons, these animals, just because you have multiple sensory channels that are sort of coming through a common neuron through a sort of common conduit? Is it just convergence? And the answer is no. So there's a whole series of studies that we've done looking at circuit dissection. I'm not going to talk about those today. Those provide a good answer to this question. Another answer to this question can be provided by the experiments that we've done looking at developing animals. And that is what I'm going to talk about today, because I want to get to talking about experience. Okay, so it is not the case that you can just jam visual and auditory inputs together and get multisensory integration. It's not simply convergence. So let's talk a little bit about multisensory development. So I'm going to show you here sort of a timeline for development in a cat. This is sensory development in the superior colliculus of cat. So cats are born with a sense of touch, but the ear canals and the eyes are closed. So hearing doesn't come online, audition doesn't come online until about 1 week of age. And then vision doesn't come online until about 3 weeks of age, give or take. Okay, so with this timeline that I'm depicting down here, this is when you can start finding neurons in the SE responsive to each of these modalities. Now, what's interesting is as soon as you start seeing multiple modalities represented in the SE, you can start to find multisensory neurons that are responsive. I'll just go ahead and talk about it, that are responsive to each one of those, to combinations of those cues. Okay, so as soon as, for example, you can find representations of somatosensation and audition, you can start to find neurons that are responsive to somatosensory inputs and auditory inputs. And as soon as you can find visual neurons, you can find neurons that are responsive to all three of these modalities. But what's crucial to know about these early appearing neurons that are represented with multisensory convergence, because they're responsive to multiple modalities, is that even though they respond to multiple modalities, their response to combinations of cues are not enhanced. So we say they're multisensory neurons, but they don't have integration capabilities. When you present spatiotemporally congruent cross-modal cues to them, right, they don't give you enhanced responses. If you then wait another couple of weeks, so between 4 and 12 weeks of age, this is when you'll start to see multisensory integration capabilities start to appear, so that by 12 weeks of age, you can find neurons that are responsive to multiple sensory modalities and give you that sort of classic bumped enhanced response. Okay, so 12 weeks of age is kind of the end, close to the end of that window. All right, so multisensory convergence here and multisensory integration are not the same thing. And this developmental plan that I just showed you is not a cat-specific thing, so you can see the same thing in monkeys and humans as well. So what's going on in the cats, the 4 to 12 week window, when we transition from multisensory convergence to multisensory integration? Well, what's going on is that the SC, and the brain as a whole, is acquiring multisensory experience. And we know that multisensory experience is necessary for this development. So how do we know that? Well, we did a number of experiments in which we precluded the animal getting multisensory experience. And we did that in a couple of different ways. So one way we did it is reared animals in a dark room. So this is not like a photography dark room, which has like a red light in it. This is a totally dark room, light tight room. You got to wear night vision goggles and all this great stuff. So animals are reared from birth in total darkness. So this blocks their visual experience, obviously, but also their visual and non-visual experience. And when you test these animals as adults, you're looking at the physiological responses of the SC, you can find visually responsive neurons. And you can find neurons responsive to visual and non-visual modalities, but they don't have integration capabilities. So you get, when you present visual and non-visual cues together in spatiotemporal congruence, the response that you get out is no better than if you just presented one of them alone. So you might look at that and you might say, yeah, okay. And if you know something about dark reading, you might say, well, dark reading does a lot of stuff. And so maybe what I'm looking at here is not the result of blocking multisensory experience per se, but maybe it's just something having to do with dark reading. So you say, okay, well, we did another technique, and this is rearing animals in an omnidirectional sound environment. So this blocks patterned auditory experience and, crucially, auditory, non-auditory multisensory experience. So this is a room that has speakers all along the perimeter, and they're pumping out broadband noise, like white noise, that's about 85 decibels at cage level. So it's not at the pain threshold, and it's you could raise animals here just fine, and it doesn't deafen them or anything like that. So these animals are not deafened by this experience. You can still find auditory responsive neurons in the SC, and you can find neurons responsive to auditory and non-auditory cues. But when you test them with the assay that we've done, right, you see that these stimuli don't elicit enhancement when presented in spatiotemporal congruence. Rather, the response that you're getting out from the neuron, the multisensory responses, are sort of no better than the unisensory responses. And so you might look at that result and you might say, okay, but in both of these cases, you were knocking out a sensory modality in order to knock out multisensory experience. You still haven't really nailed it. You still have not done the properly controlled experiment where you provide unisensory experience, but block multisensory experience exactly. So we did another experiment. And in this one, animals are reared in an arena that has LEDs and speakers around the perimetry. And the LEDs and speakers randomly activate according to Poisson processes. Okay, so they don't get spatiotemporally congruent multisensory experience. It's just kind of random activation. These animals develop actually well-developed visual and auditory receptive fields. And when you probe them with visual and auditory cues, you see no enhancement. Okay, so it really is multisensory experience per se that's allowing the animal to develop multisensory integration capabilities. So this was all SC physiology. And you might say, well, okay, that's SC physiology. Is what you're showing me at that, these are single neurons in the SC. It's what you're showing me at the single neuron level predictive in any way of the animal's behavior. Okay, so the answer is yes. And so here are some data for you. This here is a summary of the classic behavioral enhancement in that task I showed you before, just kind of collapsing across locations in a neurotypic animal. Okay, so again, this is like sort of this auditory accuracy. Here's this visual accuracy. Here's this multisensory accuracy. You see that big enhancement. This is an animal detecting, localizing spatiotemporally congruent cues. This is a neurotypic animal. Okay, you see that big bump. All right. Did the same assay in dark-reared animals. And what you're seeing here, so that line, which is labeled stat-fac, is a line that represents the best unisensory response selected on a trial-by-trial basis. So it's a sort of representation of what the animal could do if it didn't have multisensory integration capabilities, what it can do based on the unisensory information alone. Okay, so because of sort of statistical facilitation, it's slightly higher than the unisensory responses. So that's your sort of unisensory reference in each one of these cases. You see in the normally reared case, exemplified above. Dark-reared case, doesn't make it. Okay, so dark-reared animals, whole animal behavior, do not show multisensory enhancement. And this was just recently published. And then hot off the presses, we did the same thing with noise-reared animals and showed the same results. Okay, so that paper is actually being written right now. But you get the same result from the noise-reared animals as you do with the dark-reared animals. That is, you don't have behavioral enhancement. And I want to emphasize here that in these dark-reared and noise-reared cases, these animals are not blind or deaf. They have populations of visually responsive neurons. They have populations of auditory responsive neurons. They have populations of multisensory neurons that are responsive to vision and audition, right? So they've got large pools of neurons that get activated in the spatiotemporally congruent visual auditory condition, but they can't use that information to enhance their behavior like a neurotypic animal can. So it's almost as if there's like a decoder in the brain that is compromised in these animals. And I think that's perhaps why the single neuron is from the SC is predictive of these particular results, because the single neuron in the SC is compromised in these animals. So it's interesting here that the single neuron activity is predictive of the animal's whole behavior. So you might be wondering, is it permanent? All right. So these dark-reared, these noise-reared animals have defects in multisensory integration. And let me point out that, we rear animals in these conditions for experimental control reasons to test basic science hypotheses. But it's not that these manipulations are totally on Mars and different from any human condition that would ever be experienced. So, for example, people who are born with congenital cataracts have compromised pattern vision early in life and compromised visual and non-visual experience, right? And then later in life, they can get those cataracts removed, right? And they can see now. And what our colleague Brigitte Roeder has shown in a few papers is that those people have compromised visual auditory enhancement later in life. Okay, so that's sort of like dark rearing. In the case of noise ring, if you think about people who are congenitally deaf, right? So they lack early auditory experience, and then some of them make cochlear implants, and then all of a sudden they can hear, but they lack that early auditory, non-auditory experience. And what you find in some of these patients is they have compromised multisensory enhancement later in life, but it depends on how much experience they were able to get when the cochlear implant was done, et cetera. Okay. So these animal model kind of manipulations are quite relevant actually to some human conditions. And in fact, you can use the animal results that I showed you to sort of predict what happens in these different human populations as well, which is pretty cool. So if we're talking about humans, and we're talking about sort of compromised, the circumstance where we have compromised multisensory integration, then obviously the question we would have is, well, is that permanent? Or is there anything we can do? Okay, so in fact, it's not permanent. And one of the techniques that we developed, we said, okay, so if the problem with like a dark-reared or a noise-reared animal, we call these naive reared as a general category. If the problem with a naive reared animal is that they didn't get multisensory experience, maybe we could give them that multisensory experience as adults and really kind of hit them heavy with it, give them a lot of multisensory experience. So we developed what we saw as a multisensory training paradigm, which is a really boring exposure paradigm. It's called training, but there's no actual training involved. It's really just exposure. So it's sensory training, where the animal is just kind of sitting there and he repeatedly gets spatiotemporally congruent visual auditory experience. Okay, so this is spatiotemporally aligned visual and auditory cues that are repeated again and again and again and again and again. Like I said, it's maybe a little boring. So you can provide this experience in that fashion, and you can do this over. You can do this when the animal's awake. You can do this when the animal is anesthetized. You can do it every day. You can do it over the course of several weeks, et cetera. Okay. And what we found is that when we applied this to dark-reared animals, and this was done under anesthesia, when you sit dark-reared animals down, anesthetized, and you give them the sensory training experience again and again and again and again, over the course of several weeks, you can develop multisensory enhancement capabilities in SC neurons. And at the end of this, the neurons that got that experience that was presented within their receptive fields look approximately normal. And this is showing you sort of the developmental progression of that over several weeks of exposure. So you start with nothing and you go up to a high incidence of neurons with integration capabilities. And then here, this is showing you that sort of ME metric or MSI metric. Over time, you see it gets more robust as sensory training proceeds. So that's dark-reared animals, and it looks like we can use multisensory training to drive development of enhancement capabilities in the SC. What about noise-reared animals, and what about a normal environment? Okay, so one of the questions that came up here was saying, okay, so if sensory training works, I wonder if a normal environment would work. So the multisensory experience that's inherent to a normal environment is what a normally developing animal uses to develop integration capabilities. There's a lot of multisensory experience out there, even in the home environments that we have here. Okay, so we wondered, all right, sensory training, we wonder if it works in noise-reared animals, but we also wonder, would just a normal environment work? Okay, would the multisensory experience inherent in a normal environment be effective in rehabilitating these animals? So here's a sort of paradigm for this experiment. We noise-reared animals for 6 months. Then we put them in a normal environment for 6 months, and then split them in two groups. Some kept on in the normal environment. The others got the sensory training paradigm I just showed you. And what you find is that sensory training paradigm does develop enhancement capabilities in noise-reared animals, but a normal environment doesn't, not on this timeline. Okay, and that was 12 months. So that's it's a year of normal environment experience. Much longer than it takes to develop enhancement capabilities in a younger animal, right? Note that these weren't getting the experience until 6 months of age, right? Outside of the sensitive period. But the sensory training paradigm works very well. Okay. So there's something about the sensory training paradigm that's driving changes in the brain that is above and beyond in adult animals, that is above and beyond what you can get in just a normal environment. So what's required for sensory training to work? What's special about it? Well, it contains congruent multisensory experience, which, normal environments, you get a lot of variable kind of experience. And some of it's congruent and some of it's not. The sensory training paradigm is consistent and it's repeated, right? Like I said, in a normal environment, you have a lot of different types of experiences, we think, right? There's a lot more variation. And so it's maybe for that reason that the multisensory training paradigm is more effective. What is not required, perhaps surprisingly, if you are somebody who does neurorehabilitation and that sort of thing, is the animals don't have to be awake. All you need is sensory exposure, and behavior is not required, and the stimuli don't have to be associated with reward or whatever. And that's not to say that stimuli that were associated with reward or a behavioral task would not work better, right? But it's just to say that it's not strictly required. Okay. All right. So those are examples where we're taking naive reared animals and then as adults giving them sensory training to develop enhancement capabilities. And so you might look at that and you might say, well, okay, so these are kind of naive reared animals, strange animals. What about adult plasticity in a normal animal? So in a neurotypic animal, is there any adult plasticity? In the SC, multisensory plasticity? And the answer is yes. And we've done a series of studies here. The gist of this, the take-home message, is that the principles of multisensory plasticity in the SC roughly follow what we would expect from what are sometimes called temporally asymmetric Hebbian learning rules, or spike timing dependent plasticity, or STDP. So what's illustrated for you here is a study that we were doing in which we had visual and auditory cues that were like sort of asynchronous. They were very close together in time, but not precisely temporally aligned. And when you repeated this sort of slightly temporally asymmetric assembly, what you found is increases in the responsiveness to the first stimulus in that pairing, okay, which is sort of what you would predict from these temporally asymmetric Hebbian learning rules. And what the study showed, though, is that you can use, this is like our first study of it, and you can use multisensory plasticity to change the neuron's responses to the individual sensory modalities. Okay, so that got us started thinking about that. And here was another study that we did, sort of following up on that basic finding, saying, okay, now we're going to use spatiotemporally congruent cross-modal cues, visual auditory cues, present them together again and again and again in that multisensory training paradigm that I just showed to a neurotypic animal, and look to see what changes for neurotypic neurons. And what you see are increases in the sensitivity of the neuron to the individual sensory modalities, okay, in quite a few of these neurons, okay? So this is just illustrated here. This is the multisensory response. This is a visual response. This is an auditory. And so you see that before our exposure, we have this kind of response profile. We do a bunch of cross-modal exposures, like in our multisensory training paradigm. And then what you're seeing here is sort of short-term plasticity, seeing big increases in the robustness of the responses to the individual modalities. Okay, so just to summarize all of that, multisensory integration normally develops in early life based on multisensory experience, but that plasticity that you have in early life, although it extends into adulthood, although the conditions for driving that plasticity change. So a normal environment is no longer sufficient. You need sort of special training paradigms in order to elicit this plasticity. One of the consequences of those training paradigms is to amplify sensitivity of the neurons to the individual sensory channels. And so if you think about that, you might say, oh, okay, that's interesting. Is that useful in any way? So this is, very basic science kind of research, and you might be wondering, so you have a way where you can use multisensory training to increase the responsiveness of neurons to individual sensory modalities. Is there any way of using that information in a sort of translational context? So let me describe to you this project that we did, that we're doing, using what we learned from multisensory plasticity in a translational context. And this is what's called multisensory rehabilitation of Hemianopia. So this audience knows Hemianopia quite well. This is just for anybody who's a little less familiar with this. This is also called Hemianopia, also called cortical blindness. If you damage visual cortex on one side of the brain, typically people go blind on the opposite side of space, and this is what is depicted over here in this city scene. So this is unfortunately a fairly common condition. So estimates are almost like 1% of the general population over 49 years of age. The incidence here can be, if you work on a stroke ward, incidence can be as high as 50% or so of patients, stroke patients, have some sort of hemianopic condition. Now, I do want to note that a lot of those will resolve spontaneously, but quite a few do not. And so about 8 to 16% of stroke patients have long-term hemianopia of some form, most of which are sort of partial or complete hemianopia. So it's a very common sort of disorder, unfortunately, and then you can induce it by TBI and stuff like that. So these are human statistics, and there has been an animal model of hemianopia that goes all the way back to James Sprague's work, seminal work back in like the 60s. And this is a cat model. So if you lesion visual cortex on one side of the brain, and here we're talking about large lesions. We're not talking about just knocking out a portion of V1 or something. We're talking about really large lesions that are taking out visual cortex on one side of the brain. The animal will reliably go blind on the opposite side of space. And what Sprague showed in an elegant series of studies is that blindness is not just due to the cortex that's responsible for seeing now being missing. Okay, so in fact, you can resolve hemianopia. Actually, you can restore the orientation of the animal to contra-lesional visual cues by surgical interventions that are disrupting communication between the hemispheres of the brain and actually the two colliculi. Okay, so the sum, and this is kind of a series of experiments up here from Sprague and colleagues that are summarized up here on the upper right there. The summary of a lot of that work was that hemianopia is resulting, the total lack of responsiveness, total blindness to cues in contralesional space is actually reflecting a sort of competition that's taking place between the hemispheres, where when you remove unilateral cortex, you remove cortex on one side of the brain, all of a sudden the other side starts winning this competition and suppresses your ability to express the animal's ability to respond to those other contralesional cues. If you sever the sort of competition between the hemispheres, right, using a variety of different interventions, then you can restore this visual responsiveness. So we've taken that exact model and we have used it for our own purposes. And so the sort of gist of this is that you really have two kind of visual brains, so to speak. There's visual cortex, and then there's the secondary pathway involving the colliculus. And the idea we had here was that instead of doing a surgical intervention that would disrupt communication between the hemispheres to try to release the colliculus from inhibition, what if we use something like a multisensory training paradigm to sort of boost or amplify the visual input to the SC, right, and thereby perhaps be able to restore some visual processing. Okay, so the slide down here, sorry, the figure down at the bottom right there is merely indicating to you that there are these kind of multiple pathways. Just because you lose visual cortex doesn't mean you're necessarily going to be totally blind forever. You do have the secondary pathway that goes through the SC, up through pulvinar actually to cortex, and then back down to the SC. And then bottom, that figure on the left there is just showing you that the lesions we're talking about here are big lesions that compromise the LGN as well. So as a consequence of damaging visual cortex, removing visual cortex, the LGN degenerates. Okay, so it's really, these really are substantive lesions. Okay, so again, this was the idea. Can we fix hemianopia with multisensory training? The strategy here is to use our multisensory training paradigm in the blinded field. We've done this a couple of different ways. So one way in which we presented cross-modal stimuli is with a super high-tech apparatus involving a ping pong ball, where you take a ping pong ball on a stick and you knock it against the side of a wall. And so it's really, state-of-the-art technology there, but it provides a very reliable, natural visual auditory stimulus. Okay, and it's synchronized and whatever. It's a ball physically hitting a wall. And then we've also done these experiments using virtual stimuli, LEDs and speakers, like I showed you before. Okay, some of the experiments we've done are under anesthesia, and I'll point these out as I get to it. So, can we fix it with multisensory training? And it works. So this is showing you data from the ping pong ball setting, where these animals are sort of in a perimetry apparatus, sort of like this. Ping pong ball is being dropped from behind these curtains wrapped against the wall to give them this experience. And what you see is over the course of a couple of weeks, so just orient you to what's going on the right here. These are radar plots showing you where the animal is visual responsive at different tested locations. So these animals are being tested sort of in a very large portion of their visual field. So where you see green, the animal can see. Where you don't see green, the animal can't see, or it wasn't tested. So you see pre-lesion, this animal can see wide visual field, and then post-lesion, you see he's blind. So this is a right side lesion, he's blind, and left side of space. So we wait 3 months to make sure it's stable. Like I said, some of these cases spontaneously resolve in human populations. So we want to make sure we have a stable defect here. So wait 3 months, and then we start doing this training. All right, and what you see here is on this animal, day 11, you start to see responsiveness creeping back in the contralesional field. And this is being tested by whether or not he can see this ping pong ball when it's being dropped and not wrapped against the cage wall. So it's being tested with unisensory visual, modality-specific visual cues. Okay, so after day 24, if you test the animal, post-rehabilitation, you see that now he can see everywhere that he could see before. Okay, so that's an awake animal done with the sophisticated ping pong ball apparatus. Here's an anesthetized animal done with virtual stimuli, and it works too. So this animal is given weekly training sessions under anesthesia, same sort of spatiotemporally congruent presentations. It's actually only done at one location in space, but that's sufficient, right, to restore vision. It takes about 4 weeks with 600 exposures per week. And so you see after 4 weeks, you start to see these responses start creeping back. So this animal was being tested behaviorally, but he was getting the exposures when he was anesthetized. And we've done this in quite a few animals, and it works very reliably. So these are data from several animals, and you can see very similar results in each one of them. So this paradigm works very quickly and reliably. It works with, relatively speaking, few trials. It works even if you're only stimulating at a single location, and it works even under anesthesia, even when the animal's not behaving and the stimuli are not associated with reward or anything like that. So the constraints of this appear very similar to the constraints of the sensory training paradigm that was used before with the dark grid and the noise grid animals. So here's just some additional data showing you this recovery. And then here is, these are animals that are done under anesthesia, and we were testing different densities of exposures. And what you can see is that if you increase the exposures per session to 2,400, you don't really get much improvement of the recovery time over 600 exposures. But if you decrease the number of exposures per session to 100, you significantly delay how long it takes before the animal starts to show recovery. So all of these animals recovered, but at different times, and the time of recovery is indicated by the shaded region. So this paradigm is effective in restoring animals' responsiveness to contralesional cues, and we've done physiological recordings from the SC and shown that it actually restores visual responsiveness within the SC, which is silenced by the initial cortical lesion. And we did another study here, it's kind of cool, where we rehabbed an animal and then did an ibotenic acid lesion of the deep layer neurons in the SC and showed that he goes blind again. And in this particular assay, we actually did an incomplete lesion of the deep layers of the SC, sparing a portion of what would correspond to this location in the visual field, and that area was spared for the animal as well. So it really is the deep layers of the SC that are helping this animal respond to cues again. Furthermore, what I showed you is that, so in everything I showed you there, we were using spatiotemporally congruent cross-modal cues. And so what we've learned so far is that spatiotemporally congruent cross-modal cues, consistent, regular experience with spatiotemporally congruent cross-modal cues appears to be the key to driving this effect, okay, to providing this rehabilitative effect. But you might wonder whether this rehabilitation paradigm is effective because it's sort of following these same principles of multisensory plasticity, multisensory integration that I talked about before, or whether there's something else going on here. So the natural question everybody asks is, what the heck's going on here, right? So why is this working, right? So let's talk a little bit about what we learned about what specifically is required in this rehabilitative training and entertain a few alternatives as well. So one of the alternatives that people come up with, so if our position was that this is multisensory plasticity, another alternative might be that it's not really multisensory plasticity, that all that's happening here is a sort of attentional or cueing phenomenon. So when you present the visual and auditory cues together over here, the animal can hear the auditory stimulus, and that's priming or cueing him to the location of the visual cue. Okay, so when you do this again and again and again and again and again, all you're doing is kind of dragging the animal's attention over there. Okay, and we might, so if that were the case, then you might imagine that an even more effective paradigm might be one in which the auditory stimulus preceded the visual by some amount of time. Right, just like a classic priming or cueing paradigm, right? They're typically not simultaneous when the cueing stimulus happens before the target stimulus. Okay, so you might imagine an experiment to test this hypothesis in which you're going to try to rehabilitate a blinded animal with temporally asymmetric cues, such that the auditory precedes the visual by like 300 milliseconds or so. Okay, now this hypothesis would say that that would work, and then the multisensory plasticity hypothesis would say it's not going to work because it violates the temporal principle of multisensory integration. So we did this experiment, months and months of training with auditory 300 milliseconds before the visual cue in a hemianopic animal. It doesn't work. Okay. Another hypothesis you might wonder is if it's just oculomotor training. So are you really training some sort of sensory phenomenon over here, or are you just training a scanning behavior, right? Just getting the animal to like look over there, right, and sort of training this reflexive visual motor response. Well, okay, if that's the case, if that's the hypothesis, then you might say in an experiment where there's some other cue that tells the animal to look over there, right, then as long as he's looking over there and the visual stimulus is there, that should be effective in rehabilitating, right? Even if that was a spatially disparate cue, okay, that was cueing him to look over there. Whereas if you think it's multisensory principles of multisensory plasticity, you would say that that's not going to work because it's going to violate the spatial principle. So we did that experiment. So here you have auditory stimuli at 15 degrees and visual stimuli at 45 degrees. Okay, so animal's blind this side of space. All right, but the auditory cue at 15 degrees gives the animal, it tells the animal that the visual stimulus is going to be at 45 because this relationship is consistent. Okay, so what you see when you do this experiment and provide these exposures to this animal, and I should have mentioned this is awake, when you provide these exposures to this animal, is that during the exposure trials, he starts responding to that visual cue at 45 degrees because he gets a reward for going there. Okay, so we have here an animal who he's blind, based on the auditory stimulus, he learns to go to 45 degrees, which happens to be the location of the visual stimulus. And we do this for months. Again and again and again, and this profile here on the right is just showing you the animal learns to go to the location of the visual stimulus. And then we test him with visual stimuli alone, and he can't see over there. Okay, so even during training, he kept going, right? And he was rewarded for going to the location of the visual stimulus, but if the visual stimulus is presented alone, he can't see it. It's only because the auditory stimulus is telling him that there's a visual stimulus there. Okay, what does work? The same things that I showed you for the multisensory training paradigm and that we were discussing before. Congruent multisensory experience, reliable and consistent. And all those animals I've just showed you that didn't get rehabilitated were later rehabilitated using these cues. So the final question would be, okay, what can they see, right? And that's a tough question to ask an animal, but let me describe to you just very quickly a number of probes that we did. So I have shown you this kind of visual detection and localization probe, and clearly this recovers in rehabilitated animals. And I should note to you that in this testing, there are catch trials in which there's no stimulus presented and the animal's rewarded for staying still. And so he's not just guessing when he goes out there and localizes these cues, right? Because he gets rewarded for staying still if there is no stimulus there, right? And he performs very, very well in this task. So he's only going to the cues because he has some confidence that there's actually a cue there. He's not just guessing. side of space or something like that. We've done some other tests as well. So in fact, the animals can discriminate frequency gratings after they've been rehabilitated with up to a certain level. They're not as good as they were before, but they can actually discriminate different spatial frequencies using the frequency grading discrimination paradigm. And in fact, they can do some shape discrimination as well. So they can sort of tell the difference between upward and downward triangles and pluses and circles. Not as well as they could before, but they can get it above chance. And then let me describe the final task to you. And this is a visual choice task. And this is a way of assessing sort of competitiveness between the two hemifields in a rehabilitated animal. And so you might expect, even back from like Sprague's results, you might expect that even though the animal might be able to respond to cues in the formerly blinded field, that if there was a cue in the intact field, that he's not going to be able to respond to anything over there. Okay, so we did a test of this, right? So we trained animals to go to stimuli on the left or the right, right? And then we presented the cues together. These are visual cues, presented them together, and they could go to either one and get a reward. The visual choice, do whatever you want, you'll get a reward, right? So you get to see which one they pick. So a prediction here might be that if the intact side is really, really powerful, that they'll always pick the intact side, right? And so we just tested them as visual choice to see what they did. And they don't always pick the intact side. In fact, 2 out of the 3 animals who tested this way prefer the rehabilitated side, right? And this effect that I'm showing you here is not just an artifact of them preferring one side or the other or something like that. It's sensitive to stimulus intensity. So if you lower the intensity of their preferred side, the stimulus on that preferred side, they prefer it less, and you can equilibrate the responses, et cetera. Okay, so even in the presence of a stimulus in the intact field, an animal will be able to respond, will choose to respond to stimulus in the rehabilitated field. So what I've talked about today is that multisensory integration is a powerful and fundamental neural computation that significantly enhances behavior. It's a capability that's acquired from experience, typically early in life, but that experience can be acquired later in life with some latent plasticity. And the best way that we know of unlocking that plasticity is these multisensory training paradigms. And what I finally showed you there is how that plasticity can be exploited in translational context to enhance sensory processing of compromised modalities in the case of hemianopia. So here's my thanks, my acknowledgement slide. These are the people that I've been working with over some time on these various projects. These are faculty members at Wake Forest. These are the current students working very hard in the lab. And then here's current funding from NIH and a few pilot awards that are going after some of this, some new information. So thank you very much for your attention. I hope you enjoyed it, and I'm happy to entertain any questions.